there could be multiple users and so on. And finally, right, I'm just going to talk about intelligent large-scale surveillance. This is again an important problem. I mean, one can talk about lots of things, recognition, traffic management, and all that. But then the point is, right, we are going to be focusing only on roads, and especially Chennai, where I live, right, is supposed to be becoming a smart city. And one of the things that we thought we could do that could be very interesting, right, is to be able to right, bring in some amount of inference, high-level inference into the city road traffic, such as traffic jams. We should be able to report about traffic jams, accidents, crowd gathering, and so on. Maybe some kind of weak supervision that can do in order to be able to classify vehicles as bikes, autos, and cars, and so on. And of course, the key challenge is that there are going to be lots of wide intra-class variations, and then you have a high sort of a density of vehicles, right? I mean, and especially in India, and then you also have to kind of deal with occlusions and so on, right? And then of course, we would also want to extend this to the situation where it can do weather agnostic surveillance. And finally, right, I'm also happy to share with you that among international faculty, some international faculty have already expressed interest, and here are their names. Professor Chellappa from Johns Hopkins University, Ashwin Shankaranarayanan from CMU, Vishnu Budetti from MSU, and then Vishal Patel from Johns Hopkins University. With that, I'm done. Thanks for your attention.
Thank you, Raju. That was a fantastic talk, and I don't know if we can all see each other, but let's.
Thank you.
Can you? Let's see. Let me see. Maybe I can.
I'll stop sharing. Maybe I'll stop sharing my slide.
Yes.
Okay.
Yeah. All right. We see everyone. Thank you very much. It's clear your group is doing cutting edge research, and especially in certain niche areas in computer vision, deep learning and real practical problems, but very well-founded solutions. And I know you were a pioneer in depth from defocus in the late 90s, and so you really hold a key position in related fields too. Very good. And so there are a few questions that I'm seeing here in the Q&A, and I'll first go to them, and then if there is time, I can also ask some questions.
Sure.
The first one is from Mahesh Kutkodi. I'm doing a research on detection and mitigation of DOS attacks in WSNs. Is it possible to save the machine learning model trained to detect DOS and WSN practically? So I'm not clear about some of these abbreviations, but maybe you do. Could you answer that?
Unfortunately, even I do not know what DOS. I mean, I don't know. Is it like kind of? I don't know. I don't want to make a wild guess. So maybe if that particular person can come out with an explanation for what he means by WSN and DOS, maybe I can try to answer.
Yeah, that is good. The other question is, I would encourage Mahesh to go and add to his question. The second question from Mr. Krishna Narayanan is, in your computer vision research, do you use any human or animal brain models?
Not really until now, but then if you wanted to do some computer vision for medicine, like you might want to, but then at this point of time, no, we have not ventured into that at all.
Okay. There's a question from Narendra Yadama. Sir, could you please send the slides through mail? I think they like to, look at your slides. Maybe you could.
Sure. In fact, we have a YouTube channel where this entire thing is being shared. So I think that is going to be alive for a long time. So I think they can download from there. No issues.
Yeah. There's a question from Kirtan Kalaria. Can the newly proposed use of transformers for computer vision help us enable multimodal learning better?
Yes, absolutely. And in fact, some of those things that I've kind of talked about, in fact, I've already started using transformers. Yes, absolutely. It's already out there.
Okay. And the question from Krishna Narayanan, in computer vision space, what are one or two most
We are leaders in intellectual properties and have filed around 1,500 plus patents over the years. We have emerged as the country's leading technical institution in creating new knowledge, as 189 patents were filed in the financial year 2019-20. IIT Madras is also at the forefront when it comes to industrial consultancy undertaken by educational institutions in the country. All this and more has led to IIT Madras to be ranked as the topmost engineering institute by National Institute.
Madras has been privileged to host many international students from all over the world. Most of these students come for their exchange programs to either do their coursework, research work, or their thesis for a period of 6 months to 1 year.
I came here with a lot of doubts about being in a different environment, but a pleasure of being in this place. In all my courses, I found wonderful friends who helped me, and I never feel alone or being outsider.
I came here to IIT Madras researching opportunities, and I've been given the opportunity to work in testing the main sensor of the satellite. This is just one of the big projects that IIT Madras is holding nowadays, and I'm really happy to have chosen this opportunity to come here.
The best thing is you can actually know whether you are passionate about this or not, because you'll have the time and also opportunity to explore each and everything.
Let's do great things together.
Hello and good day to everyone. On behalf of the Office of Global Engagement, it is my pleasure to welcome you to the 9th webinar from the IRIS webinar series. My name is Richard George Philip, and I work as the Principal Program Administrator for IOE Initiatives at the Office of Global Engagement. From the Sensing and Vision Technology Cluster, the research initiative being presented today is named Biosensing, led by Professor Prem Bisht. Professor Prem is a professor in the Physics Department of IIT Madras. His research interests are mainly in ultra-fast laser fluorescence spectroscopy and whispering gallery modes of nano-microcavities. He has been associated with IIT Madras since 1997 after completing a JSPS fellowship at Kyoto. He has active collaborations with national and international labs. He has over 90 peer-reviewed journal publications and has delivered over 60 talks at national and international universities. Joining us as a moderator today is Professor Stephen Arnold. Professor Arnold is a university professor of physics and chemistry at the Thomas Potts Professor of Physics at NYU Poly. He has received many awards and distinctions. These include John J. Outstanding Career Accomplishments in Physics, Outstanding Publication Award of Oak Ridge National University, and Sigma C Award for Distinguished Scientific Research. Along with both of them, we also have a panel that's quite wide and from all over the world. So a special thank you to everyone who has joined here today. A quick note to the participants, please use the Q&A to enter your questions and upvote questions that interest you so that the moderator can prioritize them later on. Over to you, Professor Prem.
So I'll just share my screen. seeing it?
Yes.
Great. Center of Excellence in Biochemical Sensing and Imaging Technologies. While greeting everyone present in their respective time zones, right from good morning in Americas, good afternoon in Europe, good evening in Australia. This is Prem Bist from Physics Department of IIT Madras, Chennai, along with my team with Namaskar, Vadakkam, good evening to people in India and the subcontinent. The name of our center is, as you can see, Center of Excellence in Biochemical Sensing and Imaging Technologies. We are calling it as CEN BioSim. It's an honor to present this 15-minute webinar this evening on behalf of my 9 colleagues and their research groups here at Chennai, as seen on this page. For the students, it is important to see that the team has expertise right from the subject of physics, chemistry, biotechnology, and electrical engineering, which reveals how the modern problems are not limited to a particular subject, but require interdisciplinary approach and collaborations. In next about a dozen slides, I shall also highlight our collaborators who are in different parts of the world, as well as in India. So I would come back to this screen later. The vision of the center is given in this slide with goals, enablers, and solutions we are looking for. The goals are to look for the highly sensitive, ultra-fast, and label-free sensing and imaging along with sub-diffraction resolution. These goals are to be achieved with the enablers as whispering gallery modes, whispering gallery modes, as well as fiber Bragg gratings, fluorescence lifetime and resonance energy transfer, polaritons, fiber tips with micro electromechanical systems, and meta-endoscopy and 2D materials. We mostly deal with the size of millimeter, micrometer, and on nanometer scales. This is to be achieved with the collaboration with collaborators in India and abroad, companies, and doctors and industry partners. We have 6 tracks in this particular center. So for the first one is whispering gallery mode based sensors. We have multi-photon film FRET imaging, that's lifetime and Forster resonance energy transfer, single photon detectors and deep tissue imaging. Imaging based on bacterial stress response, optical micro electromechanical systems, MEMS, and meta surfaces for imaging and sensing, optogenetics and neural imaging, polaritons, nano-FTIR, nano-Raman based biochemical fingerprints. Again, in this slide, we give a brief overview of what comes next. For instance, here is the spectrum of 2 emitting species. We can see that it has different peaks and we can distinguish them with just their emission spectra. However, when the emission spectra overlap, let's say the blue spectrum, it's difficult to distinguish between this blue and the red spectrum now. But if we take the lifetime of this particular spectra, we can distinguish with the lifetimes clearly with the different lifetime components of this. So lifetime measurement is a more accurate way of measuring imaging the sample. So that's where the fluorescence lifetime imaging becomes important in science. Forster resonance energy transfer is another technique that works within 100 angstroms between a donor and acceptor and lifetime distributions. So imaging a biological specimen with these techniques will require hundreds of measurements with thousands of components to fit with global analysis. We need expertise in machine learning, for instance. We will see how synthetic tissues are planned to mimic the human organs. One of the most important problems of bacterial resistance to the common antibiotics is also being attended here. Some surface plasmon resonance and their hybrid systems with other phenomena help us in imaging and single molecule limits. We also have applications of fiber optics and micro electromechanical systems. We'll see these as they come along. So these whispering gallery modes are the name comes from these archaeological structures which are famous. Even in India, we have Bijapur Dome, which is even here. These photographs are from the wiki. And at the same time, if we take to the optical cavity, we have, for example, if the circumference is 2 pi r, and if the wavelength can bounce back and forth between this circumference several times, then we can have similar kind of modes, which are also known as whispering gallery modes here. The quality factor, I mean, for example, if I show you a simulation of a 6 micrometer cavity, then we can see that it has got these 90 spots, which correspond to the 45th order mode with first order, 45th number. And one of the important thing is you can see that some evanescent coupling of the light, which is coming outside the particle. The first order modes are on the periphery, but the second order modes go inside the particle. And then based upon these, the quality factor of this cavity, here is the image from the work of Vollmer and Arnold. By the way, Professor Arnold is the moderator in this talk, where the tunable laser looks at the microcavity, and the analyte comes, as it comes, there is a shift in the peak of the whispering gallery mode. And if the second analyze comes, then there is a second shift. For example, there's another photograph, another image from another paper of Arnold with my student Tantam when he was doing postdoc with Steve. And we can see that this delta lambda corresponds to the second analyte and so on. So it's possible to image these small virus type of particles with the help of these whispering gallery modes. The another geometry which we are working here is we are planning to work is the micro bottle resonator, which is shown here. There are several geometries depending upon their quality factor and other properties, they become useful. And here is the list of our collaborators, which are we are helping with us in this work. The next track is multi-photon film microscopy for single photon detectors in biomedical research. Here we can see this particular photo here shows the single photon excitation, the green fluorescence, what you see here. And here it is 2 photon excitation, which is excited by the femtosecond laser 800 nanometer. You can see red photon. And this image in the middle shows that both look similar, but the 2 photon excited, both are nonlinear optical phenomena, but one of them is Chi3 process and another is Chi2 process. They help in distinguishing the imaging of crystalline and non-crystalline tissues in biology. And with the help of this carbon-based quantum dots synthesized in the laboratory, we are also having collaboration with the Australian group of Hala Zirket with Gurvinder and Yogamba with high resolution deep tissue imaging in this up to even 3000 micrometers in hydrogel scaffold and 280 micrometers in the pig skin tissue. Related to this deep tissue imaging is work which is 3D multi-photon based volumetry imaging of engineered synthetic tissues. This particular activity is led by my colleague Shantanu Pradhan in biotechnology. And here we see that the plan is to study multifunctional plasmonic nanosystems for imaging. So here is the mouse brain, which multi-photon deep tissue image of the mouse brain. And here is the even more interesting image of multi-photon excited fluorescence of synthetic tissues, so which can mimic the human organs. This activity is also planned in this particular task. And here, of course, the FRET and FLIM activities will be also useful. And another important thing is the dynamic imaging of protein DNA assemblies governing bacterial stress response. So we know that this the bacteria, are resistance for the antibiotics recently in nowadays. And here in this particular work, we are planning for the film FRET, which is again used to look at the basic science of this proteins, and which helps in drug discovery as well as in the future work in the society, pathogenic response of these activities. So this activity is actually led by IIT Naganathan of biotechnology. And how do H and S protein, for example, molecule self-assembles and forms higher order molecular assemblies? How do these assemblies bind DNA and regulate transcription and hence infectivity? Addressing these questions has implications in various levels, ranging from basic science to drug discovery. So film-fret technique, again, is useful for imaging in vitro and in cells, as the lifetime changes are independent of the protein concentration. The next important activity is led by Shanti and Inakshi of Electrical Engineering Department on this and MEMS and meta surfaces for imaging. And what they have done is here they are looking for a micro endoscopy device, which is based upon the MEMS mirror and the optics, which is done by, let's say, meta surface, which generates airy light, so that we can have the more diffraction limited beam of light. And this is here the example of SEM image of a micro mirror for spectroscopy. So these are the examples, and the activity in this is to look for a micro endoscope with better performance. So this track 5 and 6 are led by my youngest colleague from electrical engineering, which is Shivananju with Balaji Srinivasan. In this project, we plan to develop optical devices to access the brain neurons, let's say for some animals before, and then which will be useful for the therapy to patients in neural disorders. So this figure here shows a two-dimensional graphene-based carbon electrode array, which they say is a clear device, implanted on the cortex of a mouse brain, and the blue light stimulus is being delivered via the device. And here is the angiogram showing the image of a clear micro E electrocardiogram of the brain. So the next track is also by Shivananju. The activity here is on the polaritons. We are the sulfurous plasmon resonance. For example, the single strand DNA, the gold nanoribbon coated single strand DNA is already on the prism of the Kashman geometry of the surface plasmon resonance experiment, in which the microRNA is now dropped from on top, and then we can see that the concentration ranges of 10 power 10 automolar to 10 picomolar, which are about 10 power minus 6 orders of 10 power 6 orders of magnitude sensitivity can be found from this. So these 2D material and research is being attempted in this particular track. So actually, that's all is the total research work we are doing, we are planning, and this is our international linkage on whispering gallery mode-based platforms, multi-photon film microscopy, where Professor Periaswamy in the Keck Center of Cellular Imaging, Virginia, is the expert whom we are looking for the help, as well as Professor Surinder Singh is there in this multi-photon as well as in whispering gallery mode kind of experiments. And Professor Sumit Mahajan is from Southampton, UK, and Professor Zodikasis from Australia, sorry, and Christian Hammersam from Monash University and Professor Bao from Hong Kong. And again, these people are in both of these activities. So we have some already expertise in fiber optics and MEMS, world-class facilities, as well as several experimental facilities. Some things are again being updated, augmented by under this funding. So we actually, on behalf of IIT Madras, I acknowledge the funding from Ministry of Education, Government of India under the Institute of Eminence status of IIT Madras. So thank you very much for this. And this is the team which is from IIT Madras. So that's where I would close the the presentation. Steve, would you like to?
Hey. So I very much liked your presentation. I think SN Bose would be very proud of the use of optics as it's being done right now in Madras for all these different applications. Since in a sense, the boson is the birthplace is India for the boson. So what I wanted to ask you about is how you got from your interest in fluorescent lifetimes to basically into the biosphere and trying to use this to basically detect things for biology and characterize biological materials.
Yeah, so in principle, as I explained in one of my first slides, was that the lifetime measurement is more accurate way of identifying the system if they are overlapping. For example, they could have similar spectra, but the lifetimes would immediately identify that they are different. So that was the first and foremost motivation to go for the lifetime measurements. And in these, my colleagues in biotechnology department, since they are interested in these biological proteins and synthetic tissues and imaging, therefore lifetime imaging becomes one of the important tools for imaging of such tissues.
Yeah, the surprising thing is your work, as I've followed it over the years, shows how there's a very unique interaction between cavity quantum electrodynamics and understanding of that phenomenon of the change in the lifetime. And how did you get involved with whispering gallery modes?
Yeah, so the interesting thing was that when I was doing my JSPS fellowship in Japan, one of the laboratories in Kyoto was just started this work on droplets of immiscible solvents, let's say oil water, which makes a droplet. And those are the softest spheres which we could get, but then they were not having the quality factors which are more than, let's say, 10 to the power 3, which could show any change of the cavity Q on the radiative rates. So we went for some polymer, laboratory-made polymer microspheres, which were still soft, but was hard enough to give some quality factors of 10 to the power 3 to 10 to the power 4. So that is where I started. And after joining IIT Madras, that was one of the interests, because it involves the Me resonances, which could be calculated, simulated by the Me theory. And immediately one can also measure them and correlate the theory with the experiment. So that was the idea of entering into the whispering gallery modes.
Well, I don't know if I'm supposed to ask questions of others as well.
I think so. So there must be some questions on the question answer. Balaji,
Okay.
would you be able to?
Can I comment on something?
Yeah, please go ahead.
Yeah,
Mary.
Prim, probably you can stop share so that.
Okay, sorry. Yeah, I'll stop sharing. Is that okay? Yeah.
Yeah, great.
Great.
So regarding the lifetime, Prim mentioned which I built the lifetime imaging system. I was I'm one of the pioneers in that technique. When you illuminate the light in a biological system, either cells or tissue, particularly tissue, the light has a differential absorption, meaning that, and light scattering, meaning that you will get optical artifacts. The lifetime is a quantitative technique. You are following the time response of the molecules. So that way you can avoid all the optical artifacts induced by biological system absorptions. It's a very quantitative technique, so you can use for protein-protein interaction or metabolism measurements on the clinical side on cancer. I'm working on the cancer metabolism using this lifetime technique, multi-photon film imaging systems. So in general, you can reduce the optical artifacts using the lifetime technique.
Right. So also the FRET, if Steve allows me to, yeah.
Yes, please.
Well, the FRET is again we call it as a super resolution system because you can measure the distance between the molecule within 10 nanometer. And using the previously they used the spectrophlorometry technique where you may not see the spatial information, but using the microscopy or light microscopy technique, you can get time and spatial resolution, as well as you can analyze live samples, single cell analysis. And right now we are using machine learning, as Prem mentioned, for this kind of using lifetime technique on the fast recording. For example, in the metabolism imaging, we get with the time response, hundreds, thousands of data. If I ask the student to sit and do it, he or she takes weeks or months. If you feed into the machine learning approach, you can get it within few minutes of these thousands of data of the lifetime-based metabolism measurements. Yes, the lifetime technique is a very ideal technique for FRET, which is protein-protein interactions, meaning that, again, we avoid optical artifacts. You follow only the time response of the molecules. So it's very quantitative measurements using the lifetime technique for FRET, or protein-protein interaction, either in cell or in tissue or in live animals.
Thank you.
We use all these techniques for that.
There's a question from about the computational aspects of optical sensing. It reads, "Sir, could you please explain what are the computational aspects of optical sensing?"
Yeah, I think the answer goes to this again. So I guess, Balaji, would you suggest that it should again be related to
Shantanu? Shantanu would be a good person to answer that since he does a lot of computational biology.
Yeah.
Shantanu?
Sorry, computational aspects?
Yeah, the question is, could you please explain what are the computational aspects of optical sensing?
Yeah, so I think what the answer goes back to the slide which I was showing, in which I said that when we have imaging of the biological specimen, and what just Perry was also mentioning, and to image a sample, you need to measure the sample across, and which will have about some hundreds of data sets. And each data set would have a few exponentials, their amplitudes, their standard deviations, and so on and so forth. Therefore, you finally have 1,000 data which one has to play with to make an image. And this is known as global analysis. So that's where the computational aspects, or actually machine learning, which just now Perry also mentioned, comes into the imaging. And we actually have a computer science expert, Professor Chandrasekhar, which also I sent an invitation today. He is in our proposal where we will take help from him.
There's also a question about what are the possible ways of reducing artifacts in neural imaging using computer vision techniques, reducing artifacts in neural imaging.
Yeah, so I think Shivananju may give some idea on the neural imaging.
I think the question is on the computational, but I can talk about something on the optogenetics, what we are planning. Actually, most of the neurons will be controlled by electrical impulse. So the idea of our project is to introduce some kind of biomolecules from the algae or the protein, use some gene therapy inside that molecule into the brain. So the molecule will have kind of a photosensitive response, assume that around 475 nanometer. So we want to stimulate that particular biomolecules or neuron by stimulating with light, so that when we incident the light onto the biomolecule will induce some electrical pulse. Based on that, we can activate the particular neurons. So this has some huge potential application for neural disorder. That's our project on this optogenetics and neural imaging. We are also planning to build a kind of a compact microscope so that along with the optical fiber, where we can also image these neurons along with stimulant. So we are using the graphene electrode. So why graphene? Because if you take indium tin oxide, that's the material they use. So comparing to indium tin oxide, graphene is a better material. The main reason is flexible, because since it's a flexible, I can easily implant on the brain surface. And also it's 90% transmission. Actually, it's a 97% transmission, and there is around 2.73% of absorption. And the uniqueness of graphene is, since it's a zero band gap material, it can absorb a broadband light, like it covers most of the spectrum, so that we can use any light source to stimulate these neurons. So it has various applications. That's a clear device which we are planning to build using this COE project, so that we can implant on the neurons. So we'll be stimulating the neurons with light and recording the signal by a clear device.
Say a question about what is the general sensitivity and specificity of these optical biosensing techniques. Maybe I can handle some of that. I had the pleasure of having a former student from Madras as a postdoc in my laboratory, and he was the first one to actually do single biological molecule detection ever with these whispering gallery modes, without the need for any labeling of the biological molecule, but rather by using specific antibodies on the surface of the resonator, which was looking at these things. And that involved using a plasmonic enhancement of the whispering gallery mode biosensors. So all of this sort of thing is the sort of thing that is done in Prem's lab. And I think it is incredibly sensitive. So I hope I've answered that question. Someone's writing, the lifetime community is limited compared to intensity-based insights like neural firing super resolution techniques at present. What could be the future important projects of lifetime in biomedical image processing? I think we've had a little of the answer to that. But do you want to?
Something related to, so lifetimes are more accurate way of
right.
imaging. And if something is not fluorescent, then they are labeled by some fluorescent tagging systems, and accordingly the images are produced.
Right. So it probably should be pointed out, even in the case of NMR imaging, the precession of the proton spin, you measure, in a sense, the relaxation time. And that's the way you make an image of an organ, the brain, whatever it is. So that could be an analog of the same sort of thing, but that doesn't use fluorescence, of course. So
actually, lifetime, the future is you can use it for non-fluorescent molecule for NADH, which is autofluorescence.
Right.
It is a kind of, we are using clinical side, prostate cancer, for example. It's if you follow the NADH, which is a metabolism mitochondrial, it is available in the mitochondria, NADH molecule. It is autofluorescence. Right now, the lifetime technique is kind of bench to bed. We are using directly to the clinical side, taking prostate cancer tissue and patient samples to test the NADH level and then compare to the PSA, which we use for all the time for PSA prostate specific antigen testing, PSA testing for the prostate cancer. We do that all the time in the clinical side. But using lifetime technique, you can able to follow the metabolic activity by following the mitochondrial dysfunction. In the case of prostate cancer, that is where the lifetime technique is advantageous because, as I mentioned before, less optical artifacts, and you can follow quantitatively the information from the clinical samples. That's,
yeah,
I mean, we are leaning towards that side, clinical side.
Someone's asking the question, what type of reporter molecules or fluorophores are used for FLIM-FRET?
Yeah, Perry, actually you can attend that. Any molecule you
you can use, currently they are using biosensor for Venus and any red fluorophore combination. They are using for biosensors experiment for many clinical applications for particularly screening the molecule using biosensors, biosensor FRET. That is very actively involved in the clinical side. There are a number of visible fluorescence proteins, which is GFP, Venus, M-cherry. There are a number of fluorophore molecules now available which has a high quantum efficiency. For example, Venus has 0.6, and so high quantum efficiency is required for donor molecule to get a better FRET. So when they develop the biosensors, they do look for a quantum efficiency of the fluorophore molecule to develop the biosensor to use for molecular screening.
Thank you.
And there's a question here, but I don't understand it. It says, which electrode is used in the biosensor?
So all what we are talking were the electrodes, but in the sense that probing things, all the techniques are electrodes.
And that seems to.
I have some question, if you permit me.
Sure.
To ask our endoscopy team, especially from so even IIT. So how are we, making more advancements in this particular endoscopy scheme?
So I'll take that, Prem, thanks. So we're looking specifically at developing micro endoscopes. There's been a lot of work on that of late. And again, endoscopy, as most people will be familiar with, is a tool for imaging within the human body. But developments now are not just to do regular imaging, but to use all the techniques that have been discussed here to employ those along with endoscopy. So how do you do 2-photon microscopic? You use the 2-photon techniques and use that along with endoscopy. So there's research going on in that. But the